आज तुम लोग लेट पे आए हो. Try to come in time. So welcome to the third class of nanoscale modeling and simulation. In the last class, we were talking about what is the content of the course or what we are going to do in the course, and we take the name and highlight on modeling, and we told that we keep few questions, we model nanostructure, and also what do we mean by modeling something. I was trying to answer what do we mean by modeling something. Modeling something is categorically divided in two parts. One is designing and another is development. What is designing? Designing means change or modify according to our requirement for something which is not exactly needed to be developed. It is there, but we are modifying it according to the requirement of our planning, requirement of our experiment. Let's say we have a nanoparticle, but now we want the nanoparticle to be interacted with a surface which is positively in charge. So we definitely develop the surface being negatively charged, right? The opposite charge. Similarly, the it would be in the reverse way. Also, if something is hydrophobic, we make the surface hydrophobic. If we require magnetic property, then we take magnetic nanoparticle. In that way, with the and we will talk about that with the requirement of the experiment, we will design according to that. Another one is development. Development is building something from scratch. You don't have something, you have to develop it, or assembling subunits into a meaningful structure. The second one is more appropriately applicable to biological macromolecules. So in this course, what we used to do, we try to learn both the things. We try to learn designing, we try to learn development. As an example of development, I used to talk about the modeling of macromolecule, especially protein, because protein modeling is a very classic example of modeling. And by understanding protein modeling, you would understand a very useful and a very futuristic nanoparticle which we are going to take in future. So keeping that in mind, we want to perform the modeling of biological molecule protein, but at the same time, because people come from various backgrounds, to give them a certain introduction, we plan to talk about biological macromolecules so that you could get the basic knowledge about biological macromolecules. And I have started that with central dogma of life, because when you want to understand biology, it is very important to understand how biology is running. Biology runs through the main three processes which together call the central dogma of life. What is that? We talked about that DNA, by the process of replication, from another DNA. DNA by the process of transcription from mRNA or messenger RNA, and messenger RNA by the process of translation from protein. Here we have two types of macromolecules. DNA and RNA are nucleic acids, and protein is developed by amino acids. Amino acids are the building block. So are there other macromolecules here? We have started defining the four major classes of biological macromolecules where we end in the last class. One is carbohydrate, and this picture gives you a basic introduction to the type of carbohydrates commonly present in food. Protein, again, the picture gives you an introduction of common type of foods which include in the category of protein. Fat, again, the foods are there. And last but not the least, the nucleic acids in which ribonucleic acid and deoxyribonucleic acids are included, which are RNA and DNA. We have already talked about them. Now going into little details which brings us in carbohydrate. Carbohydrates serve as the fuel and building material. It includes sugars and polymers of sugar. So the basic unit of carbohydrate is sugar, and sugar polymerized to more complex sugars. The simplest carbohydrates are monosaccharides or single sugars. Carbohydrate macromolecules are polysaccharides, polymer composed of many sugar building blocks. So if you see, you will see there, they are connected with the oxygens and they form polymer. It could be 100,000, which is denoted by the number n. As you see here, this is a straight chain polymer. It is not restricted of the polymer being straight chain. It could be branched chain also. Here you are looking at an example of branched chain polysaccharides or polymer of sugars. So monosaccharides, generally they have a unique molecular formula which is multiples of CH2O. So this is CH2O whole X. So there is carbon, there is water, that means hydrated carbon, and the term carbohydrate comes from there. So carbohydrate have a unique or common formula of CH2O whole X. Glucose, which is CH2O whole 6 or C6H12O6, is the most common monosaccharide. Wherever you get, food, you will find the presence of glucose there in very high amount. It is the most common one. Monosaccharides are classified by the location of the carbonyl group. If you are going to take a look, you will see that monosaccharides have carbonyl groups, and depending on the position of carbonyl group, they are classified. Also, how many number of carbons in the carbon skeleton. So if you see here, this is 6 carbon, so it's called hexose. It's 5 carbon, it's called pentose in that way. If you look at here, they could be cyclic form, they could be in open chain form. There is 3 carbon, so it is called triose. If it is 1, 2, 3, 4, 5 carbon, it is called pentose. So glyceraldehyde is an example of triose, and ribose. If you remember, in DNA and RNA there is ribose and deoxyribose. They are example of pentose, and then there is hexose, which is glucose and other related one like fructose. Coming to fat, they are constructed from 2 types of smaller molecule. One is glycerol. If you remember the triose, we have already looked at. Triose is, glycerol is an example of triose, and fatty acids. So glycerol, as we have seen already, is a 3 carbon alcohol with a hydroxyl group attached to each carbon. If you see, there is 3 carbon and each of the carbon is attached with 3 hydroxyl group. So this is called glycerol. Fatty acid consists of a carboxyl group attached to a long carbon skeleton. So if you see all of them, there are different fatty acids like acid, lauric acid. Okay, if you look at the examples, you will get linoleic acid and all those things. The commonality here is it a long fatty acid chain, long chain of carbon, which is ended up with acid. So it is a carbon chain followed by a acid group. Everything have that. Now all these would be replaced to make a develop a ester linkage CH2O in the fatty acid, and when three of the alcohol hydroxyl group are replaced by three fatty acid chain to form ester, they are together called fat or lipid. There are two type of fats. One is saturated fat and another is unsaturated fat. You see here, butter is an example of saturated fat, and here you could see the glycerol moiety, then oxygen, the ester link, and the fatty acid, and three of them are straight, which means the chains are saturated. Because they are straight, it's a very interesting thing I'm talking about. You could find that with a small experiment in your daily life, when the fat molecule have saturated fatty acids. What I mean by saturated fatty acids, all of them are single chain. They are straight. They are straight, and because of that, they pack very nicely. Because they pack very nicely, the movement of those fat molecule are relatively less, and that brings them in low kinetic energy, and with lowering of temperature they are even more well packed, so they become solidified. Whereas if you look at unsaturated fat like olive oil, you could have heard about virgin olive oil. So if you look at in virgin olive oil, it have the same structure. Only difference is two chains are really same like this, but the third chain you see the introduction of a double bond. Because of the presence of this very double bond, it is called unsaturated fat. When there is a double bond, there is unsaturation. When there is no double bond, there is saturation. Okay, because of the presence of this double bond, they have a kink. Because here the double bond means, when there is a double bond, it could stay in same side, which is called cis conformation, or in opposite side, which is called trans conformation. You will see the kink because it is in the same side, means it is in the cis conformation. Because of that, this is not developing as regular shape. To understand this better, like here, it's like this, it will create a bend. Because of that bending, it could not go and pack better, and because of the because of its inability to pack better, it is kind of shows fluidity, that means high kinetic energy, so lowering the temperature don't usually make them solidified. So if you imagine in your mind now, you get like butter is an example of saturated fat. Coconut oil is an example of saturated fat. Olive oil is an example of unsaturated fat. Okay. The oils which are example of saturated fat would be solidified at lower with the lowering of temperature. The oils which are unsaturated, especially mono-unsaturated, they would not be solidified with the lowering of the temperature. So, structural formula of a saturated fat molecule is here. You should also look at the space filling model of the stearic acid, which is example of a saturated fatty acid. Here you see the structural formula of an unsaturated fat molecule. This is the space filling model. But now what I'm going to talk about is even more interesting. I would talk about unsaturated and polyunsaturated. When you see the monounsaturated, you see there is a clear bending. But when there will be multiple unsaturation, they would balance and ultimately they would get a shape more like this. So, saturated fat is not good. Unsaturated, which is monounsaturated fat, is good. Polyunsaturated fat is again not good. And that's the reason, if you see, when you go to market, your elders who are having heart problem look for oil which have high proportion or high quantity of monosaturated fatty acid. Olive oil have 60 to 80% of monounsaturated fatty acid, and which is virgin olive oil that have nearly 80% of monounsaturated fatty acid. And because of that, if you see, olive oil is very, very costly and very, very good for health. I hope you will understand this. You could anytime ask me question if you are not sure. Then we come to phospholipid. Phospholipids are like fat, but here in the tail there is a phosphate which is followed by a hydrophilic head. If it is positive, it is positive in nature. If it is negative, it shows negative in nature. So instead of three, the first two are like fat, but the third one is different. In phospholipid, it have a phosphate and a head. It might be positive or negative, but it's generally create a hydrophilic head. Here it is choline. So phosphatidylcholine. So it have ultimately hydrophobic tail and hydrophilic head. So in a phospholipid, two fatty acids and a phosphate group are attached to glycerol. The two fatty acid tails are hydrophobic, but the phosphate group and its attachments form a hydrophilic head. Another one is called steroid. In the steroid, it is characteristics with 4 fused ring. 3 of them are hexagonal and the fourth one is 5-membered. The steroids are also lipids characterized by a carbon skeleton consisting of 4 fused rings. One of the very popular example of steroid is cholesterol. It is an important steroid. It is a component in animal cell membrane. So cholesterol is essential. So everybody needs cholesterol, but when there is excessive cholesterol, what happens? Excessive cholesterol, if you see, this is a rigid body, so it again reduces the fluidity, and when the fluidity of the membrane reduces, as you have seen in case of saturated and polyunsaturated fatty acids, it like kind of lose its dynamicity, and that leads to heart disease, that leads to stroke and all those things. So although cholesterol is essential in animals, high level in the blood may contribute to cardiovascular disease. Coming to, so this is about carbohydrate and fat. Coming to nucleic acid. Nucleic acids are polymers called polynucleotides. Each polynucleotide is made of monomers called nucleotides. Each nucleotide consists of a nitrogenous base, a pentose sugar, and one or more phosphate groups. The portion of a nucleotide without the phosphate group is called a nucleoside. This is the cartoon of a double-stranded DNA. As we have talked about, there is a component sugar and nitrogenous base and phosphate. The sugar could be deoxyribose, like not having a hydroxyl group here and only a hydroxyl group here, and not having a hydroxyl group at position, which defines the sugar for DNA. Whereas when both the C2, C3 positions will be occupied by hydroxyl group, that is called the sugar for RNA. There are different nitrogenous base: adenine, guanine, cytosine, and thymine. If you look at, and there is uracil for RNA, adenine, guanine are purines which have 5-membered ring followed by 6-membered ring. So there is a 6-membered ring and a 5-membered ring. So it's a bigger structure with a smaller name, purine. And cytosine, thymine, and uracil, they are 3 with only 1 ring. They are smaller, so they called by a bigger name, pyrimidine. So purines are 6-membered followed by 5-membered ring. Pyrimidines are only 6-membered ring. I hope this will help you remember. Then coming to the fourth one, the protein. Proteins include a diversity of structures resulting in a wide range of function. Proteins account for more than 50% of the dry mass of the most cell. Proteins' functions include structural support, storage, transport, cellular communication, movement, and defense against foreign substances. But these are only few examples. We will take some examples, but proteins are the molecules which are performing the function. There are 2 types of proteins. One which build up your structure, they call structural protein. Like a nose, it looks like a nose and not like an ear because of the structural proteins. So the proteins which develop our structure are called structural protein. Protein which help us do our function are called functional proteins. So there are 2 types of major category proteins: structural protein and functional protein. The functional proteins are also called enzymes, and you all know enzymes are very important molecules. Our big part of discussion will include the enzymes. So here are a few examples to define protein function. Enzymatic proteins, which I was talking about, they do selective acceleration of chemical reaction, like A plus B to C plus D. This reaction, if you have to perform in the laboratory, you have to provide some external factor like heat, like temperature, pressure, sorry, heat and temperature is same, pressure and anything else. But That's not possible inside the body, right? You don't take, kacha chawal, like the rice molecule, take some water and sit on the oven and it, boils inside. It never happens like that, right? So the reactions which cannot form easily in physiological condition, enzymes help them happening. How enzymes help them happening would be a real good amount of discussions which would be coming in the following classes. Defensive proteins, protein which helps in protection against disease. For example, antibodies inactivate and help destroy viruses and bacteria. As you guys all now know, we are in COVID era. You are sitting home, you are taking vaccines. The vaccines will help your antibodies to be activated. Storage protein, the function is storage of amino acids. For example, casein is the amino acid which is found in milk, is a major source of amino acids for baby mammals. Plants have storage protein in their seeds. Ovalbumin is the protein of egg white used as an amino acid source for the developing embryo. Transport protein, they help in transportation of substances. Example is hemoglobin, the iron containing protein of vertebrate. Blood transports oxygen from the lungs to other parts of the body. Other protein transport molecules across cell membrane. So in the body there are a lot of transporting happening, and transport protein take specific role in carrying them or transporting them. Talking about protein, as I told, it is very important. The building blocks of proteins are amino acids. They are called amino acids because if you see there is a amino group, there is a acid group. That's why they call amino acid. If you look at the general structure of the amino acids, you will see there is amino group, as I told, acid group, one hydrogen, and then there is one R. To make it easy, you could imagine R as X. As in mathematics, X means a variable. Here also R means a variable. When the R is H, this is the simplest amino acid. This is called glycine. When the R is CH3, it is called alanine. In that way, by changing R, 20 different R means 20 different essential amino acids. They are histidine, arginine, lysine, isoleucine, phenylalanine, leucine, tryptophan, alanine, methionine, proline, cysteine, asparagine, valine, glycine, serine, glutamine, tyrosine, and aspartic acid, glutamic acid, and threonine. So these are the 20 amino acids which builds up the most essential and most fundamental functional biological macromolecule, which is called protein. Okay. Amino acids histidine, arginine, and lysine are basic in nature. Phenylalanine, alanine, leucine, methionine, isoleucine, tryptophan, proline, and valine are kind of neutral, hydrophobic in nature. Cysteine, glycine, glutamine, asparagine, serine, tyrosine, and threonine are polar, hydrophilic in nature. Aspartate and glutamate are negative in nature. Protein, as I told, the amino acids, they stitches with each other. That is called the primary structure. Then from primary structure, they develop hydrogen bonds and they form alpha helix and beta sheets. Alpha helix and beta sheets are connected by loops. They are called the secondary structure. From secondary structure, they further fold, that called tertiary structure. So from one polymer, you go up to tertiary structure. But if you have multiple monomers, they come together. If two monomer comes together, it's called dimer. If three comes together, it's called trimer. If four comes together, as you see here, two orange and two green, it's called tetramer in that way. So there is very interesting part to look at. Every protein have a unique three-dimensional structure, and three-dimensional structure of such proteins would help us understanding what would be the function of that protein. But there are many more interesting parts which we are going to talk about. One is when one amino acid connect with other amino acid, what they form is called peptide bond. And you could see the peptide bond. The peptide bond have this phi and psi, you see, dihedral angles. We will again talk more about this, but this is where you could have understand the basic of the modeling development rules of the protein. If protein does not have those things, we could not talk about the development of protein model. What I'm trying to say, there is C double bond ONH. So in a peptide, it is C double bond ONH. So is it single bond or double bond? My question to you guys. C double bond ONH, between C and N, it is a single bond or a double bond?
Single bond, sir.
Name?
Asmita.
Anybody else?
Hello, single bond, sir.
Louder.
Yes, sir, single bond.
First your name, na?
Udit,
Yeah. Is it that difficult to make that decorum? Yeah, please do that. So yes, apparently it looks like a single bond, but there is a resonance because you see C double bond ONH. If you look at N, and I would be little more descriptive on the next class, nitrogen habits lone pair of electron. It could provide that lone pair of electron, oxygen become O minus. So one resonating structure of peptide represents a double bond, and that's why it is called that the peptide is empowered with a partial double bond. Is that clear to everyone? Everyone understand why the CN bond is a double bond?
yes,
Yes,
sir.
sir.
Now, what is the difference if it is a single bond or a double bond? In a single bond, it could freely rotate, right? Is this rotation possible in a double bond?
No, sir.
No, right?
Yes, sir. No.
Everyone, please try this. Can you do the rotation?
No, sir. It's not possible.
Yeah. So because with the introduction of the double bond, the freedom of free rotation restrict. That gives you a power to predict. You understand what I mean? A power to predict which conformations are allowed and which conformations are not. And again, I'm saying I will go into details because that is the basis of modeling, but you would be really, really surprised and proud to know that for different amino acids, they have different size, so they have different number of allowance of the conformation, right? They could rotate different proportion, right? If they are bigger, you could rotate less. If you are smaller, you could rotate more. Do you understand this?
Yes, sir.
Now, in 1960s, when computer was not developed up to a certain level, by using the crude apparatus, by crude apparatus I mean the tools you have in your geometry box in when you are in 10 plus 2, an Indian scientist group by the able leadership of Professor G.N. Ramachandran in India, using the model made of thermocol, have predicted how the allowance would happen and how from that allowance we could have plot the phi and size. So he make a plot of pi minus 180 to plus 180, psi minus 180 to plus 180, and plot the allowance of these phi and psi angles in a peptide bond. And today we are sitting in the era of supercomputing. No scientists have come up with a better method to calculate this. This is called Ramachandran plot. I will talk more about that, but again, just to summarize, the C double bond O in H is the connective bond between two amino acids, and it should be a single bond. Then there will be a free rotation, and that free rotation makes or allows protein to adopt millions and billions of conformations. Interestingly, that does not happen. The nitrogen with a lone pair develop a resonance structure, giving it a state where it is showing its double bond nature, partial double bond nature. Because of the partial double bond nature, it restricts the rotation of the amino acids around, and definitely the restriction is different for different amino acid depending on the size of the amino acid. But that's not surprising. Surprising is by looking at those changes with the crude instruments, Ramachandran and his group come up with a plot which is obeyed by any natural protein produced by Mother Nature. And not only that, that gives you the standard so that today, when people are designing novel protein, the ultimate standard they are looking for is Ramachandran plot. The protein is only validated to be going to theoretical designing to experiment when the conformer of the protein obeys Ramachandran plot. I would be happy if you have any question in this point. This is the most critical point to understand the protein folding, the basis of protein modeling, development of a model. See, you have to understand, see, you are in a very interesting scenario where in one side you are looking at the designing of a modeling, and in other side you are looking at a development. Here we are developing the concept of the development. So I would be happy if you have any question.
Hello, sir.
Yeah, little louder.
Sir, why it's very difficult to
I cannot hear you.
why it's very difficult to predict the position of proline in Ramachandran plot?
Sorry.
Why it's very difficult to predict the position of proline amino acid in the Ramachandran plot?
So did I talked about that?
No.
Then everything have a perspective. I will give you answer, but only one thing I appreciate any and every question, be it silly, be it smart. I don't mind, but I don't like people to show over smartness. I'm sorry, because to me, the person who know already is not the only student in the class. You probably don't have idea, because as you have already asked this question, so I am telling you, here there are three people sitting named Advaita, Jyoti and Riya. They are master in protein, but still they are not talking like that, because they know in my class they are not the important ones. Everybody is important. It is my goal, though no one is a backbencher, but to educate the course to the person who finds the course the most difficult. So don't ask questions which I have not developed, because that's what development is all about. If today I talk about proline already, then why proline come, how proline come, those would not be answered. I never, offend with any questions. But please understand, the class is for everyone. I could have, like go to more smarter things, but the goal is to reach to everyone, talk to everyone, make it understandable to everyone. As a teacher, that's my duty. I hope you understand, right? So, guys, anytime you ask questions, I will be happy. Please ask questions, because we are not in one room. So I want to reach you. I want to, help you understanding this. And this is something, very interesting, but very different. So if you ask questions, you actually could help others. Coming to my favorite topic, where I name it chemistry meet biology. See, the entire biological world, so big, right? But you would be surprised to see that the entire biological world is developed with hydrogen, carbon, nitrogen, oxygen, 95% or even more than that, and phosphorus and sulfur, rest of that. So only these six elements in the periodic table, you know about them, and you know about biology. That's a very interesting part, right? So let's talk about them. Elements in biological macromolecules. Carbon, it can form vast number and variety of compounds, you all know already. Vital to organic and life processes. Carbon is the base of organic and life processes. It can form four covalent bonds. It could often seen in covalent interaction in hydrophobic regions. Nitrogen is critical for many biological molecules. Ammonia, often the starting compound for many nitrogen compounds. It can form three covalent bonds, often seen in covalent or hydrogen bonded interaction. Non-covalent. Oxygen, very reactive, combines with most elements, component of many organic compounds. It can form two covalent bonds. Hydrogen binds to carbon, nitrogen, oxygen atoms, not very reactive, can form one covalent bond, participate in hydrogen bonds. Hydrogen bond is so famous, so effective that it name in the name of hydrogen. Phosphorus, very reactive, key element in energy currency in biology, can form 3 to or 5 covalent bonds. Sulfur, similar to oxygen, found in two essential amino acids, can form two covalent bonds. So this is what the basic about the biological macromolecules. We have talked about who are they, what are their building blocks, how they form, how they function, how they work, how they categorize, all of this. In the next class, we will talk about their reading, how we could read them. And more interestingly, we will look at the further proceeding regarding them, how they could be known and how that information would be used, which one we should read, which one not, and all these things. So I will finish here and I would love to see if you have any questions. Thank you. Any questions? Yes. Any question?
No, sir.
Okay. Thank you then. See you in the next class. Bye.
Thank you, sir.
Thank you, sir.
Thank you, sir.